3 species of Opihi in the main Hawaiian Islands. The Blackfoot, the Yellowfoot, and Ko'ele, or the kneecap Opihi. And this is something that's been in the news a fair bit, is that people are aware of the fact that Opihi are now the most expensive seafood in the state of Hawaii. The idea is that Opihi fishery has crashed. If we looked at historical values for how much Opihi harvest there was close to the 1900s, it ranged anywhere from 75 to about 150,000 pounds per year. Since 1940 monitoring, we've got sales that are on the order of 8,000 or so. This is the onset of a size harvesting regulation in the state of Hawaii, and you can see that that harvest regulation has not changed that trend at all. This is data from commercial sales for the commercial harvest, and as of today, that is the only data that we have for Opihi abundances. One of the potential answers for looking at reversing that decline is marine protected areas. So this is something that Ross touched on in his talk. The idea of an MPA is that there's zero harvesting allowed in that zone. Organisms are able to live out their lives in that particular area without direct interference from humans. And the idea is that those individuals in that area can grow to a larger size. They can grow old and spawn many times. And as Ross mentioned, that for fish, we don't know if this is true for Opihi, but at least for fish, the larger an individual is, the greater its mass-specific reproductive output. And so an MPA would be successful if it produces enough offspring that it then can sustain the surrounding unprotected areas for additional harvest. And so the idea would be that we have Opihi, we want to protect a couple of areas where we're going to allow those individuals to breed. We can then harvest other areas, and even if those areas are harvested to extinction, there are protected areas where there are individuals that can reproduce. And over time, as we have spawning, we get recruitment and eventually adults in those surrounding areas again to enable further harvest for a sustainable fishery. And the reason that we have to understand something about population boundaries for this is, in the same scenario, if we actually have two different populations that we don't really know about, we get spawning and recruitment, and this population on this side gets none of the recruits, and they're harvested to extinction, and they stay that way, while this group here has enabled continued harvest. Well, one way to do this is to tag individuals and to find out where they move. And so people who are familiar with Carl Meyer and Kim Holland's work, you can take a tracker and you can stick it in a fish and you can see where it goes. Unfortunately, when you stick a tracker this big on an Opihi larva, they don't move very far. So these things, the only way that we can figure out how something that is a fraction of a millimeter in diameter is moving is to use some indirect tool such as genetics. And so we want to try and use DNA to try and figure out what, if any, population boundaries exist within the Hawaiian archipelago. And if we want to manage these populations, how do we go about doing that? Well, the way that we might use DNA is that we're going to sample one population. We look at the frequency of the genetic variants in that population. We look at a second population and show that those two have the exact same genetic frequency of individuals. And we'll assume that those belong to the same population. We look at a third location, and it has a very different distribution of genetic variants in that third population. We assume that those things are not exchanging migrants on a regular basis that is going to be sustainable for fisheries harvest. So if the population genetics between two locations is very different, that suggests that exchange between those areas is rare enough that it's not going to provide a subsidy for management uses. So in terms of what we know about this, this is work that was previously funded by Sea Grant that sort of set the stage for this project. Chris Bird has already published this work. What we find for Ko'ele is that we see a huge break that the island of Kauai shares no genetic variants. The haplotypes are completely different. There are no shared haplotypes with anywhere else in the main Hawaiian Islands. And that it varies by species. So if you're looking at the Blackfoot, there are some breaks that are different than Yellowfoot Opihi and so on. And so the island of Molokai was showing some differences in the initial data, and we decided that we would focus our next study on that and spread out. So we have the previous study based on mtDNA that uses the traditional experimental design. We go out one time, we pick up the adults, we take those adults, we sample them, we run the DNA, and we look at structure. But that tells us something about structure on the scales of hundreds to thousands of generations. It's not telling us about individual dispersal events. So the design of the current study that we are undertaking is a very specific age-structured sampling. So we're going out, we're collecting the adults that are spawning and producing the offspring. We're collecting a bunch of new recruits that are coming in at those sites, and we're assigning those recruits back to the populations of adults that produce them. So then we can get at ecological timescales. We can actually start to talk about dispersal events and where are larvae coming from on ecologically relevant scales that matter to managers.